Welcome to day 3 of the reverse transcriptase PCR lab. So very quickly, just to go over what we did on days 1 and 2. Originally what we did is we isolated some mRNA from mouse liver, and we used an enzyme called reverse transcriptase, and what it does is it takes mRNA and converts it back into cDNA. The trick there, though, is that the new DNA doesn't have any of the introns that were in the original DNA. All of the mRNA is made of exons, so what we have is a piece of what we call cDNA. It's not naturally occurring DNA like in the mouse, but instead a piece of DNA we synthesized that is made up completely of exons and does not contain any introns. So we made a whole bunch of copies of all the different mRNAs that are found in the mouse liver. But that's not what we wanted. We were particularly trying to isolate the enzyme GAPDH. And GAPDH is used in glucose metabolism. And we picked GAPDH just because it's convenient. The mouse liver is constantly undergoing glucose metabolism, and so we know that it's most likely to have the GAPDH gene because it is regularly using the GAPDH enzyme. So GAPDH was chosen to study in this particular reaction because of convenience. So on day 2, what we did is we took all of the cDNA that we had, and we didn't want to amplify everything. All that we really wanted to amplify was our GAPDH section, or gene. So we created some primers and put the primers just before the GAPDH gene. And we added some Taq polymerase, and we did PCR. And PCR occurred and made a whole bunch of copies of just our GAPDH section. And we put that in the fridge until day 3. So now on day 3, what we're going to do is we're going to run a gel to make sure that what we got is actually what we're expecting. So we're going to pour the gel just like we pour every other gel. We're going to use the Mini 1 gel electrophoresis apparatus. You're going to take the comb out of the gel, place the gel in the carrier and pour the buffer solution over it, and then you have to prepare your samples. So every tube that you get gets 12 mils of your PCR master mix solution, which now has, hopefully, millions and millions of copies of the amplified GAPDH gene. And you're also going to add some loading dye. This is that purplish looking stuff, and what that does is keep it from floating out of the well. It adds some nice density to it. So we have 6 combs in our gel. Our gel looks something like this. And we have 6 wells, and all of these lanes are numbered, so 1, 2, 3, 4, 5, 6. So we're going to have the ladder, group 1, group 1, group 2, group 2, and another ladder. And the only reason we put ladders on both ends is to make it a little easier to read. If you only have a ladder in lane 1, the group that has their sample in lane 5 might have a difficult time reading the ladder. So you can run 2 ladders just to make it a little easier on yourself. So once we load 10 microliters of these solutions into each of the wells, we're going to turn the gel electrophoresis apparatus on, and that'll need to run for about 15 or 20 minutes. And what we should hopefully see, you can see down here, we put the ladder that we're going to use. We know that the GAPDH gene is approximately 1,000 base pairs, so ignore what I'm highlighting here. It should be 1,000, not 500 base pairs. So with our ladder, we should be looking for the second bright line. So here's a really bright line. Here's the second bright line at 1 kilobase, which our gene should be that size. So what we should do is we're going to look at our ladder, and we look for the really bright lines. I'm going to use red. Then we'll get a few more bands and another bright line. And a few more, and then finally our third bright band. And the bottom band would be about 0.5 kilobases. And hopefully what we would see is one band representing our gene at the correct size. And of course the other ladder would run very similar on the opposite side. And that's what you're expecting to see here. You're looking for some DNA glowing fragments that are about 1,000 base pairs long. I hope it works out for you.